Welcome back to Chapter 4, the three-dimensional structure of proteins. In this lecture, we look at part 3 of this chapter, which deals with protein tertiary and quaternary structures. In addition to looking at protein structures, we'll also look at how proteins fold and unfold in solution. Before I begin my discussion on protein tertiary and quaternary structures, I want you to understand one more concept on protein secondary structures. Now, this deals with a technique called circular dichroism, which is a method to analyze the secondary structure of a protein or a peptide. Any form of structural asymmetry in a molecule gives rise to differences in absorption of left-handed versus right-handed circularly polarized light. Now, you have to remember that proteins are chiral because amino acids are chiral. And hence, any changes in the secondary structural elements will give rise to a change in circularly polarized light. Measurements of this difference in circularly polarized light is called circular dichroism spectroscopy. Circular dichroism spectroscopy, or CD, measures the molar absorption difference, that is delta E, of left and right-handed circularly polarized light. So the difference is equal to EL minus ER. Now, what contributes to this change in molar absorption is the chromophores. Remember, proteins have chromophores, at least 3 of them. 1 is phenylalanine, 2 is tyrosine, and 3 is tryptophan. Chromophores in the chiral environment produce characteristic signals. CD signals from peptide bonds depend on the chain conformation. Very important. Shown here is an example of a CD spectra, and this is a CD spectra of polylysine. Polylysine is a polymer of lysine residues. It can adopt 3 different conformations depending on which solvent it is dissolved in. Now remember, in the previous slide I told you that chromophores in proteins come from 3 aromatic amino acids. Polylysine does not contain an aromatic amino acid. So, where does the absorption come from? For proteins and peptides, spectra are obtained in the far UV region, and this is called as a far UV region between 190 and 250 nanometers. The light absorbing entity or chromophore in this region is the peptide bond, the CONH bond. It's basically the backbone. And a signal is obtained when the peptide bond is in a folded environment. The difference in molar extinction coefficient for, like I said, the left-hand and the right-handed CD, or the circularly polarized light, is plotted as a function of wavelength. So delta E, there is a difference in the circularly polarized light between the left and the right hand, right, is plotted versus the wavelength. And you will get CD spectra for 3 different secondary structures. For an alpha helix, it looks like this. Now, as you can see from this graph, there is also positive and negative values. For alpha helix, it has what is called as a minima, and minima close to 210 nanometers and 225 nanometers. A beta conformation will have a graph like this, whereas a random coil would look like this. Using CD spectra, biochemists can determine whether proteins are properly folded. They can estimate the fraction of protein that is folded in either of the common secondary structures, and also monitor transitions between folded and unfolded states. So remember, this is a very important experiment that determines whether a protein is in a folded state or in an unfolded state, and that is dictated by the secondary structural elements. Let us begin our discussion on protein tertiary structure. Tertiary structure refers to the overall spatial arrangement of atoms in a protein. It is stabilized by numerous weak interactions between amino acid side chains. Largely hydrophobic and polar interactions are responsible for making tertiary structures. They can also be stabilized by disulfide bonds. Remember, I was talking about disulfide bonds. Two sulfurs attaching to each other, that makes a disulfide bond. Usually two cysteine amino acid residues form disulfide bonds. Interacting amino acids are not necessarily next to each other in the primary sequence. Remember that. There are three different classes of protein superstructures. The first class is called as fibrous proteins. These are protein subunits arranged in long strands, mostly alpha helices. Examples include keratin and collagen. Globular proteins are proteins arranged in any orientation. There is no specific orientation involved. Mixtures of secondary structures are usually involved in forming tertiary structures. Membrane proteins are integral or peripheral proteins. Integral means these membrane proteins are embedded in the membrane. Peripheral means they are associated with the membrane. Usually helix bundles and beta barrels are involved in forming membrane proteins. They contain hydrophobic and hydrophilic regions. Usually hydrophobic when they interact with the membranes, like cell membranes, and hydrophilic regions is hanging out of the membrane so that they can interact with water. Fibrous proteins share properties that give strength and or flexibility to the structures in which they occur. In each case, the fundamental structural unit in a simple repeating element of secondary structure is what matters. For example, all fibrous proteins are insoluble in water, a property that is conferred by a high concentration of hydrophobic amino acids in both the interior and the exterior of these fibrous proteins. Now, if you look at this table, there are three different columns. First is the structure, then the characteristics, and then the examples of occurrence. The first case, alpha helix, cross-linked by disulfide bonds. Characteristics of such a structure is it's very tough, insoluble, protective structures of varying hardness and flexibility. Examples are alpha keratin of hair, feathers, nails. These all have this alpha helix cross-linked by disulfide structure. Beta conformation, soft, flexible filaments like silk fibroin. Collagen triple helix has high tensile strength without stretch. Some of the examples are collagen of tendons and bone matrix. So these are the examples of fibrous proteins. Let us consider the structure of alpha keratin in hair. Hair alpha keratin is an elongated alpha helix with somewhat thicker elements near the amino and carboxy termini. Pairs of these helices are interwound in a left-handed sense to form two-chain coiled coils. This is a two-chain coiled coil. These then combine in a higher order structures called protofilaments and protofibrils. As you can see, these two-chain coiled coil then combine together, and when they combine together, these are held together by various weak interactions. Remember that. Protofilament or protofibrils. Now, about 4 protofibrils, that is about 32 strands of alpha keratin in all combine to form an intermediate filament. And what does an intermediate filament look like? And that is how it looked like, right? Now, this is a cross-section of a hair, and within the cross-section of a hair, you have alpha keratin and two-chain coiled coil, and combine together to form a protofilament and a protofibril. Now, these individual two-chain coiled coil in various substructures also seem to be interwound, but the handedness of the interwinding and other structural details are not clear. However, a hair is an array of many alpha keratin filaments and made up of substructures as shown here. And the basic secondary structure is an alpha helix, which combine with another alpha helix to form a two-chain coiled coil. Remember this. Collagen is an important constituent of connective tissues such as tendons, cartilage, bones, cornea, etc. Each collagen chain is a long glycine and proline-rich left-handed helix, the rare exception. Remember, in the previous part, I told you that no proteins are known with left-handed helices. Collagen is one of the rarest of the rare examples which has glycine and proline in an alpha helix, and hence the alpha helix happened to take a left-handed form rather than a right-handed form. Chains consist mostly of repeat sequence where gly, X, Y. X is often proline and Y often another amino acid called 4-hydroxyproline. If you remember chapter 3, I told you about post-translational modifications, right? Once protein is expressed, those amino acids can be modified into amino acids that are different from the 20 naturally occurring amino acids. And one such example is 4-hydroxyproline. It also contains special links between collagen triple helices using modified lysine residues. 3 collagen chains intertwine into a right-handed superhelical triple helix. Shown here is the structure of collagen. The alpha chain of collagen has a repeating secondary structure unique to this protein. The repeating tripeptide is shown here. It's a glycine, proline, and 4-hydroxyproline. This adopts a left-handed helical structure with 3 residues per turn. The repeating sequence used to generate this model is what it is. Like I said, the glycine, proline, and 4-hydroxyproline. 3 of these helices, as shown here, in white, blue, and dark blue, wrap around one another with a right-handed twist. So each helix is a left-handed helix. When these 3 left-handed helix combine together, they wrap around to form a right-handed twist. The 3-stranded collagen superhelix shown from one end in a ball and stick representation is what is shown here. Now, when you show in this representation, right, you have to understand that when you show the ball and stick, you're showing individual atoms of amino acids. And it is essential, in some case, to look at the individual atoms. So, when you look at this, you can see as to how this right-handed twist is different from an individual helix. And that is what this representation is here to show. In addition, it also tells you there's very little room for van der Waals radii. That means that they're very closely packed. The 4-hydroxyproline residue in collagen is structurally different from proline because it forces the proline ring from a C-endo conformation to a C-exo conformation. When you add the hydroxy, endo conformation changes to an exo conformation. The advantage is that it offers more hydrogen bonds between the three strands of collagen. As I said before, this involves a post-translational processing and is catalyzed by prolyl hydroxylase and requires alpha-ketoglutarate, molecular oxygen, and ascorbate or vitamin C. So why is this discussion important? Why do we have to talk about prolyl hydroxylase or 4-hydroxyproline? And that has to do with vitamin C. Because vitamin C or ascorbate is involved in making 4-hydroxyproline. And why is vitamin C important? Because its deficiency causes scurvy. Scurvy is caused by vitamin C deficiency. And vitamin C is required for many things. And one of them is hydroxylation of proline and lysine in collagen. Scurvy is a deficiency disease that is characterized by general degeneration of connective tissues. So, what happens when you have scurvy? Manifestations of advanced scurvy include numerous small hemorrhages caused by fragile blood vessels because you do not have strong connective tissues and the blood vessels becomes fragile. You can have tooth loss, poor wound healing and reopening of old wounds, bone pain and degeneration, and eventually heart failure. So scurvy can be deadly. Now, the question is, how does ascorbate or ascorbic acid or vitamin C helps in preventing scurvy, right? That has to do with this chemistry here. Prolyl hydroxylase, the enzyme, converts prolines to 4-hydroxyproline. And in this process, the enzyme uses alpha-ketoglutarate and molecular oxygen in presence of iron in +2 state. And this results in succinate and carbon dioxide. Now, at times, what can happen is prolyl hydroxylase can decarboxylate alpha-ketoglutarate into succinate by converting Fe2 plus to Fe3 plus. Now, if Fe2 plus gets converted to Fe3 plus, the prolyl hydroxylase enzyme is dead and not functional anymore because Fe3 plus is not active. So to prevent this, ascorbate gets involved in the reaction of converting alpha-ketoglutarate to succinate and gets converted to dehydroxyascorbate, thereby preventing Fe2 plus getting converted to Fe3 plus. So it essentially acts as a suicide substrate for the enzyme prolyl hydroxylase. Now, because this happens, Fe2 plus stays active and your proline residues get converted to 4-hydroxyproline in collagen, and your collagens are strong. This is the reason why ascorbate is important. Collagen fibrils are collagen superstructures that are formed by cross-linking of collagen triple helices to form collagen fibrils. So each of these structures, these individual ones, are collagen triple helices, and they are cross-linked to each of them via covalent bonds. And covalent bonds between lysine or hydroxylysine or histidine amino acid residues is what these are. And if you look at the two-dimensional structure, you can see individual triple helices, and these are individual triple helices if you look at the section of these structures, and they are cross-linked. And this entire structure is called as collagen fibrils.